So, welcome back. In the previous lecture I gave some idea about the measurement, why measurement is important and what kind of which species is of our interest, I mean measurand, which property in this one. So, this means once you have defined the species that to be monitored, we have to define which property of that species has to be monitored. Then I talked about importance of the measurements, what are the criteria that a measurement has. It has to has three important things. It has to be descriptive, selective and objective. These are the three very important criteria. Then one, if you want to do some qualitative analysis, and then I talked about that there are two types of measurement. One is qualitative measurements and other is quantitative measurements. So, qualitative measurements, we talk about the data, and in the qualitative measurements we do not have data, we just give some information like high, low, good, bad, perception or something like that. So, you are grading a very good, for example, grading systems, you have A, B, C, D, but A, B, C, D also is defined that okay, you have marks in this range. So, that is actually some sort of semi-qualitative it becomes. And then we talk about the active and passive measurements. So, measurements can be of two types, one can be active measurements and other can be passive measurements. So, in the active measurements the measurand itself has the active information, it has some sort of information and which information we try to measure, whereas in the passive information the measurand does not have the measuring property. So, what we have to do, we have to give some kind of excitation or we have to give some information to the measurand and then that can be measured. That is sometimes called exciters. For example, in atomic absorption spectroscopy, in the solution you directly cannot measure the concentration. What you have to do, you have to ignite, you have to make it, you have to energize this one, and then bring the samples in the gaseous form, different atoms are isolated, and then you put a light on that one. So, this when you are putting the light, this means it is a kind of passive information, passive measurements, it is not an active measurements. Okay. For a measurement there are three important criteria, it has to be descriptive, selective and third one was objective. Now, apart from for any measurand, these three properties should be there. Another selective, when we talk about the selectiveness, here selectiveness becomes the species of interest. Which one we want to measure, okay. So, in this species of measurement, what we will be using? We will be using one of its physical property that we want to measure, okay. For example, we want to measure concentration of calcium in a solution and we want to use atomic absorption spectroscopy. So, in the solution form you have calcium, okay, and what you are going to measure? You have in the solution you will be measuring the absorbance due to calcium, okay, we have to measure this one. And then you have to again here you have to use reference, we will discuss that one, okay, or standard, we most of the time in these cases we call standard, okay. Now, when you do this one, there are very important criteria that your sample must be homogeneous, okay. This you have to first you have to actually find a representative. So, the sample is representative itself, sample is representative, and once you ascertain that sample is my sample is representative, I can go ahead with this sample, then you have to ensure that your sample is homogeneous. If your sample is not homogeneous, then your results could be biased. For example, if you are collecting a sample, so say for example, this is actually field and this is a soil that is there actually, and here you have a like some drainage is there or something is coming. If you are collecting sample from here, if you are collecting sample from here, here and here, this sample will be having very different value compared to others. So your sample is not homogeneous, okay. Your sample has to be homogeneous. If there is a not homogeneous, then you have to make it homogeneous. You have to collect the samples from all the locations, mix it and then do this one. And especially in the case of solid there is a problem, but especially in the case of gaseous and liquid samples there is no problem. Most of the time the samples is homogeneous. Of course, you have to ascertain that there are actually heterogeneous solution as well. So if you have heterogeneous solution, you can't do anything. The solution I am talking about the homogeneous. So if you are, it is in the gas form or in the liquid form, then actually there is a kind of sample is representative, it is homogeneous. Solid samples, if you are having a solid sample, for example, if I give an example here, representativeness, if you are taking a blood sample, okay, and we want to do some test on your blood samples, is it necessary that your sample should be taken from the hand or or any part of the body actually where the blood is flowing? The blood composition is same, so it can be used from anywhere. This can be taken from this place, or here, or here, or here, or even in your chest, or your thighs, or your legs, anywhere the blood veins is there, you can collect the sample, because the blood is actually supposed to be homogeneous, okay. But for example, if you have to test your brain activity, then you have to collect samples from here. Now imagine a situation that you have some problem, okay, and you have developed a lymph nodes, and the lymph nodes are, in the lymph node there will be some secretion of some chemicals there. So if you are measuring the secretion, that one, if you are collecting samples from that places, it might be actually different, okay. So this you have to be very careful in this one, that when you are choosing the sample. In the solid samples there is actually not heterogeneity, but what you do, you take a random small sample from there and from a bulk sample, and that will be actually will be a representative, but you have to actually you have to ensure that the sample that you collected, that is bulk, is actually like representative or homogeneous. Solid samples, in the previous one we talked about with no heterogeneity, okay. For example, you are talking about the sugar, or you are talking about the wheat flour that we are eating, actually there is actually not much much heterogeneity is there. If you are taking a tablets, the tablets are actually, there is not much heterogeneity in that one. So the tablets, medical tablets, they are usually homogeneous. Then if you are talking about the samples, solid samples, which obviously has heterogeneity, okay. So in that case, you have to take samples from each homogeneous regions. For example, here I told, from each homogeneous region you have to take, and then mix these samples in the same proportion. You have to remember this one again, because the proportion has to be same as between each region of the sample and a whole. So you have to ensure this one as well. If it is a suspected but not certain that a bulk sample is heterogeneous, then it is necessary to grind the samples. You have to grind it, make a fine powder, and then actually thoroughly mix it before making a random sample from the bulk. So this things that you have to actually do, and this is what is happening. This is also very important when we are doing the air pollution monitoring. What is happening? You have to get a representative samples, and if you are not getting a representative samples, for example, you are collecting the samples very close to the source region, the concentration will obviously be biased, it will be higher concentration. So you have to, there are actually strict guidelines, we will come to that one, that where you have to collect, what height you have to collect, and other things, okay. So if your sample is not homogeneous, you usually do kind of preparation and extraction also. Sometimes what is happening, your samples may be present in a very small quantity, then what you will do, in that case you have to concentrate the sample. For example, you use rotary evaporator, okay, and then there are actually like there are maybe more than one analytes are present in a sample. Then in that case, what you have to do, you have to separate them actually. So for example, if you use column, okay, so sort of column chromatography, for example, you have to do. So in this one, the column has a specific property, and based on the retention times of the eluent, they will come at different times. So you know that this particular analyte is coming at this time, okay. For example, here, so if I just put it actually this axis, and if I put it here time, okay, and here analyte, okay. So this will be what will happening at time t1, analyte 1 will be coming, okay, at t2 analyte 2 will be coming, at t3 analyte A3 is coming, and so on. So you know this one, and then what you have to do, you have to, if you have to measure A3, you have to ensure that you are measuring at this time period. So all the samples are actually ruled out, they are not measured, only sample that is coming out at T3, that will be measured, okay. So this is how the importance of the separation is, okay. And then for example, when you have to do concentration, maybe very small amount is present of the analytes, then you have to do concentration, need to concentrate these before analyzing, okay. Heavy metals in animal tissues, if you want to do, for example, if you want to measure heavy metals in animal tissues, because the metal concentration is very low, there are two options, that you take a huge number of, you take large samples, so basically high mass R, and then actually you do the processing sample preparation, and then you collect the heavy metal and measure. The second option is that you do the extraction, and then you pre-concentrate the analyte that you are interested in measuring, okay. Additives, for example, additives in polymers, so they will be also will be in very small quantity. Herbicides residue in flour, okay, if you want to measure, some herbicides in that one, then the concentration is very low, so you have to concentrate in that one. And this bit actually may be very helpful to concentrate complex mixtures selectively. So you don't concentrate all the things, you concentrate the complex mixture that you have to selectively concentrate, not concentrate all the solution. If you are concentrating all the solutions, then also it is not good. So selectively you have to select that one, that okay, I am interested in this analytes. You have to ensure that you are actually first separating that one by whatever means you are doing, and then you are actually doing the kind of analysis. And most of the pre-treatment is actually like extraction, okay. So extraction is very common pre-treatment process. And this is one of the example of the extraction that I have given here. This is actually called classical extraction method, and this is Soxhlet extraction method. What is there? Your sample is there, okay. Yeah, so this is a flask that actually is like organic solvent, okay, and you have, so what this will actually this will be heated, and this will be actually like when it is heated, it will increase, it will become volatile, okay, and you have sample in porous thimble, in this thimble you have sample, okay. And then what will happen, when you heat this one, the vapor will come from here, okay, and then solvent vapor is going like this, it goes like this, okay, and from that side on the top you have a condenser. So you actually, condenser means basically you are doing cooling. So when you are doing cooling, what will happen, this analyte, that water vapor that is generated here, it will actually fall drop by drop here actually, and because you have sample here, okay, and in these samples this will be start dissolving. So, sample you have in the porous sample, exhaustive reflex for 1 to 2 days, this is very expensive, very time consuming, because this in order to have your sample is somewhere, you have samples and in this one you have to extract. So, if you are directly putting, maybe some of the deeper samples are the samples that are there, they are not completely extracted, dissolved in the solution, and then solution of the analytes in the volatile liquids, for example, either dichloro-DCM, dichloromethane, or you use chloroform, whatever kind of things you are using, okay. And then you have, once you have collected this one, you take it out and you evaporate to the dryness, you have to completely evaporate it until the samples become dry, and then dryness or suitable concentration for separation analysis. So, this is how you do in the Salk slate extraction. So, here your sample was, for example, there is this sample was actually in the form of solid, okay. Usually, for example, say we have aerosol sample, okay. So, there is a huge heterogeneity in the aerosol samples if you are collecting for the PAS measurements. So, what you do for PAS measurements, you have to basically dissolve this one, this aerosol particles you have to dissolve in the organic solvent, and then you have to measure this pHs by GCMS or GCA, whatever the methods you have. I think I will skip this one, method of analysis, but just give you, there are two methods, one is classical and other is an instrumental. Again, and I talk in the classical methods, you can have qualitative and quantitative both, identification by color, indicator or boiling points, order, etc. So, just qualitative information that something is there, the analyte is present, but it does not give the information about how much it is present. And then quantitative, like mass or volume. So, you have gravimetric or volumetric concentration. Here actually you know that how much mass or volume is present. And then the instrumental method, second one actually qualitative, like chromatography or electrophoresis they are actually used to identify measuring physical properties, such as actually spectroscopy or electrode potential you do. A compound absorbs at a particular wavelength, so you do measurement at that particular wavelength, okay. The same thing, this can become qualitative if you know that if you are measuring a specific property of the analyte, this is very important. So, you have to first thing you have to ensure that which analyte you are interested. So, this becomes your species of interest, and then a specific property, this is actually your measurand. Which property, as I said, that there may be more than one property that can give rise to some concentration, but one can contaminate but other cannot. So, you have to do which measurement, which measurand you have to use this one, okay. And then you have to determine the relationship between the concentration, okay. So, that measurand or the property, because this is a kind of some sort of physical or chemical property that we will be having in next lecture, and then that property must have a relationship in terms of concentration. So, it has to have a relationship, the property must have a relationship with the concentration. This means whether the property measure which we are going to measure, that should increase with the increasing concentration or that should decrease. So, but there should be a relation. If there is no relation, then it becomes you cannot measure this one, okay. And often the same instrumental method is used for qualitative and quantitative analysis, okay. So, here the instrumental method, if I say you have one classical method, in the classical methods qualitative and quantitative you do not have much of the things here, not much instruments are involved, not complicated instruments are involved, but here you can have the same instruments that can be used for measuring qualitative and quantitative analysis. For example, you use a spectrophotometer, and if you are measuring, you can measure the absorbance, okay. So, once using the spectrophotometer you can measure the absorbance, and this measurance, this absorbance will give that a particular analyte is present, okay. But if you just do kind of reference and standard, if you measure the relationship between absorbance and concentration, and draw a kind of calibration curve. We will discuss these things in future lectures, that if you can establish that relationship between absorbance and concentration, then you can actually do the measure, you can measure the, you can provide the concentration of the compound in the solution. Okay. For example, there are different types of instrumentation. What property, for example, if you are using emission spectroscopy, fluorescence you are using, phosphorescence you are using, or luminescence you are using. Here what is the property? Actually here we are measuring emission, and emission because of the emission from the compound or the atom or molecule that we are actually doing this one. Okay, so when it is radiated, it will give some emission signal. Similarly, radiation absorption you can have. Okay, absorption spectroscopy, photometry, then you have NMR, ESR. Okay, you have ESR. Here the point is that here what you are measuring? Absorption you are measuring. So here the physical property is actually like is absorption, and what you will measure here? We will measure the absorbance. You are not going to measure. This is the physical property. In the last lecture I hope that I have made it very clear. This is the physical property. Okay, and this is the quantitative that we want to measure this one. So this is quantitative one. So you don't measure absorption, you measure absorption. So it is a process, but the value that how much absorption is there, it is quantified through the absorbance. Okay, so you do the absorbance here, and then for example it's diffraction. Some of the particles can diffract, do the diffraction process. So this is again a physical process. Radiation, when the sunlight is light is passed on their surfaces. Okay, so they can diffract the light in different directions. So for example you have X-ray and electron X-ray. Okay, then you can do rotation. Radiation can be rotated, and this is actually very important, and very polarimetry and circular dichroism. So this is here actually you measure the degree of polarization that we measure actually. Okay, then in some of the cases, for example electric potential is measured. This is actually again a physical property, and then you measure in the potentiometry titrations, you measure the electric potential. Okay, so and then electric potential has a direct relationship with the concentration. Then some cases you measure electric current, such as voltammetry, amperometry and polarography. So here you measure current actually directly. Electrical resistance, this is very important, conductometry, and one of the very important instruments, for example ion chromatography, uses this one actually, electrical resistance. So you do this one. Then for example if you are also measuring this kind of presence of kind of how much is the ions, then you measure the conductance of the solution. Okay, for example even in the water heavy, you just do these measurements and then get the ratios. Then mass to charge ratio, this becomes very important, especially in the mass spectrometry. In the radioactivity we measure activation, okay, or isotope dilution methods. So this is actually measure that how much radioactivity or activity a particular radioactive isotopes has, and then we try to infer the information from that one. One of the very example I show here is actually, for example, is the measurement of absorption by spectrophotometry. What you see here, so here you have analytes in the matrix that is there, okay, and this one you have a simulated light, energy simulators. Until and unless you have energy simulators, you cannot measure analytes here. And then the analytical response, you will get some analytical response, especially if it is absorption, then you will be measuring this absorbance. Okay, so this is an analytical response. Okay, so this response is collected in the form of data. This is actually is encoded information. So the example, for example, I am giving here is a spectrophotometry. The instruments, for example, there are different things here. The nomenclature that we use, the instrument is a spectrophotometer. Stimulus is monochromatic light energy. So this is actually single wavelength light energy that we are putting. If you don't put this one, you cannot measure here. And then analytical response or measurement is light absorption. And then how do we report this value? We report this value as absorbance. There are actually transducer, then data is usually represented in terms of electrical current. You have a data processor current meter, reading is meter scale, and then data domain, the way encoding analytical response is electrical or non-electrical signal. So this is actually a different kind of other part of this one, but any kind of measurements that we'll have is like you have a simulator here, and then this simulator excites this one or absorb the particular radiation, and then you have analytical response, that is actually detector we call it. Okay, so that has to detect this analytical response, and that analytical response has to be detected. These detectors may not may directly provide the information, or it may provide some other information which may be actually converted to the data or the other kind of thing. So here kind of same thing is given about the light intensity of a photocell, and this it gives a current. So you measure the current by current meter, and then you do the scaling in this one. So generally there are three components that becomes very important here. Detector in general, it device that indicates measures change in the environment. So detector is actually that is final value that will be giving. So the detector response has to be, this will tell that whether the particular light is present or not, as well as this will also, if you just further couple it, then it can also provide that how much absorption is there. Transducer, we use transducer to convert non-electrical signals to electrical data, because most of the time this becomes very important. You have to you can collect this one electrical data. Collecting the electrical data is very important here, and it's very what do you call, it's less time consuming, and this gives most of the places we record the data in either in the terms of where we get non-electrical data, we convert the data into the electrical data. And then sensors, there are different sensors. That sensor is a device that converts any physical chemical data to a electrical data, and then electrical data is converted to the measurement. We will discuss these things in next few lectures, because then when we talk about the data quality and then assurance. So I will stop here, and if you have any question and comments, please do write to me and we can discuss those things. I hope I have given two very introductory lectures on these topics about the measurement, and then properties of the measurement and lights, why the pre-treatment is required for any kind of measurements, why we need extraction, what are the different component of a measurement. So instruments, I gave very simple example of the spectrophotometer, because mostly we use that one, this is very common. But you can, of course, you can complicate things. That is not part of more complication. Instrumentations are not part of this course, so we will discuss sometimes later when we get time. So I'll stop here, and just suggest that keep reading. I have given one example. You can now, with that example, you can put how many instruments, whatever the instruments that you are going to use, you can convert that one, that what are kind of things that are needed in that instruments. Okay, so thank you very much. Bye bye, take care.